On the rocky seafloor off the European coast, a crab goes about its business, totally unaware that it is no longer in charge of its body. It stopped being in charge years ago, when something tiny crept in through a small hole in its exoskeleton and took control. The crab is alive, but has been made a puppet. It will never reproduce, it will never molt again, and when it cradles a mass of eggs, the eggs it is protecting are not its own. They belong to Sacculina carcini, a barnacle, although not a barnacle you would recognize. It has no shell, no legs, no recognizable body plan whatsoever. It has transcended the need for an independent body. It is a zombifying parasite that has dissolved the boundary between itself and its host. These are the Rhizocephala. The rhizocephalans are strange, and ordinary barnacles are already weird. Normal barnacles belong to the crustacean class Cirripedia, and they look like white volcano-shaped shells cemented to piers and boats or whale skin. These are the thoracican barnacles, filter feeders that spend their adult lives glued in place, kicking food into their mouths with their feathery legs. Here is what a normal barnacle crustacean looks like as a baby and what they look like inside their shell as an adult. But rhizocephalans are something else entirely, a parasitic group that has abandoned every recognizable feature of the crustacean body plan in the adult stage. The adult female rhizocephalan consists essentially of two parts: a reproductive body hanging out as a visible sac on the exterior of the host, and a branching internal network of threads throughout the host's body, like roots of a small plant. The root-like network is in fact where the group gets its name, rhizo meaning root, and cephala meaning head. The Rhizocephala have over 260 described species, and they parasitize a stunning variety of crustacean hosts: crabs, shrimps, hermit crabs, anomurans, and even other parasitic crustaceans. The life of a rhizocephalan begins, somewhat deceptively, as something normal. Like all crustaceans, they start as a nauplius larva, tiny, helmet-shaped, free-swimming specks equipped with 3 pairs of limbs. Recent work revealed that rhizocephalan nauplii are in many ways more anatomically detailed than their free-living barnacle relatives, preserving ancestral features that thoracican barnacles have lost. Rhizocephalan nauplii do not feed. They carry with them a yolk supply sufficient for several molts and use this time purely to develop, cycling through a series of instars with a single goal: to become a cypress larva. The cypress is a crustacean larval form unique to the Cirripedia and is its settlement stage, or the stage responsible for finding and attaching to a suitable substrate, which in the case of free-living barnacles means a rock or a shell or another animal. But in the case of the rhizocephalans, means a living crustacean host. The rhizocephalan cypress is sexually differentiated. Male and female cyprids have distinct roles and fates so different from one another that describing them requires treating them almost as separate organisms. The female cypress larvae are responsible for initiating infection of the host. These tiny larvae swim through the water, detecting chemical cues emanating from potential hosts, and when a suitable one is found, the cypress settles onto the surface of the host's exoskeleton. The cypress molts into an intermediate stage called the ketragon, a tiny cell mass equipped with a hollow stylet, a biological hypodermic needle made of chitin. The ketragon drives this stylet through the soft membrane at the base of a seta and injects a mass of cells directly into the crab's body cavity. This injection is the entire animal. Once the injected cells have reached their destination, the process of establishing the internal network begins. It starts as a small mass of undifferentiated cells but grows with extraordinary invasiveness, sending branching filaments outward to the host's body. These rootlets follow the host's tissues, insinuating themselves along the gut, around the genitals, into the hepatopancreas, and into the nerve cord. The network extracts nutrients directly from the host tissues. It is a merger of parasite and host at a level that makes removal essentially impossible. Eventually, a bud develops and pushes outward to the host's body wall, emerging on the underside of the crab's abdomen, precisely the location where the female crab would carry her own eggs. This external structure is the reproductive body of the parasite. In Saculina carcini, it appears as a smooth yellowish sac, roughly kidney-shaped, sitting where the brood mass should be. It is the only part of the adult female rhizocephalin that is visible to the outside world. The adult female Saculina, considered as a whole organism, including both the internal and external elements, has no defined body shape in the conventional sense. It has no clear body axis, barely any defined organ systems, no boundary that meaningfully separates outside from inside in the way that an epithelium normally would for most animals. It is a network, and its shape is the shape of its host's interior. When the parasite first emerges as a sac from the host, it is not yet sexually mature. It must be fertilized, and this is where the males enter the story. Male rhizocephalins do not grow up. They never develop into free-living adults. The male cypress larva settles onto the host crab, but on the external sac of an already established female. Once there, it enters a structure on the surface called the male receptor, a small pore, and injects its own cells just like the female cypress once injected itself into the crab. The injected male cells migrate to a specific location inside the female and grow there. What they grow into is essentially a testis. The adult male rhizocephalin is little more than a gamete-producing tissue mass living inside the female's body. Some species have multiple males residing inside a single female simultaneously. Male rhizocephalins are among the most reduced animals known, existing solely to fertilize eggs, housed within the body of a female, who is herself housed within the body of a crab. After fertilization, the sac fills with eggs, and the host begins to behave in ways to help along their parasitic children. Female crabs with egg masses perform a stereotyped set of behaviors to aerate and protect their brood. They climb to elevated positions, wave their abdomens to circulate water over the eggs, and perform a characteristic pumping motion. Crabs infected with Saculina perform all of these same behaviors, with the same timing and intensity. The parasite has co-opted the full suite of maternal behaviors, redirecting them entirely towards rhizocephalin reproduction. This behavioral manipulation is one of the most studied aspects of rhizocephalin biology, and it is extraordinary for its specificity. It is not a general disruption of behavior, as the infected crabs are otherwise totally normal in their locomotion, feeding, and predator avoidance. The manipulation is targeted. The mechanism is believed to involve interference with the crab's hormones governing the reproductive cycle and brood care behavior. The rhizocephalin is in direct contact with the host tissues, and it is thought to either produce hormones mimicking those of a female crab, or to interfere with the normal hormone signaling in ways that produce the same effect. The manipulation extends further. When a male crab is infected by Saculina, the parasite does not simply sit inside the male body, it feminizes it. Infected male crabs undergo physiological changes that shift them towards a female hormone profile, and in some cases their external morphology shifts as well. Male crabs can also perform the same brood care behavior as infected females. In another act of manipulation, rhizocephalins suppress molting in their hosts almost entirely. An infected crab may never molt again after the parasite is established. This serves the parasite's interests. A molting crab would shed the external parts of the parasite along with the rest of its exoskeleton, and the internal parts would lose their anchor. By blocking the molt cycle, the parasite ensures its own continuity, but the cost to the host is significant. Without the ability to molt, it cannot grow, cannot regenerate, and it is permanently locked into the body it had at time of infection. The reproductive output of the parasite is extreme. A single sac can produce tens of thousands of nauplius larvae per brood, and the parasite is capable of repeated brooding over its lifetime, with new clutches maturing after each brood is released. The host crab diligently fans each clutch until the nauplii hatch and swim away into the water column. These larvae then begin the cycle again. Even if the external sac is lost, new ones can be formed. The ecological consequences of rhizocephalan parasitism ripple outward well beyond the individual. Sacculina infection can reach rates of up to 50% in some wild crab populations, and because infected crabs are removed from the gene pool, the parasite can exert significant pressure on host populations. An infected crab is not dead. It may live for years after infection, continuing to feed and occupy space in the habitat, but it contributes nothing reproductively to the host population, while simultaneously investing its resources in parasite reproduction. The intimacy of the relationship between Sacculina and its host raises genuinely difficult questions about where one organism ends and another begins. The parasite is in such close integration with the host that some researchers have started using language of chimera to describe the infected crab, a single biological unit composed of two genetically distinct organisms. The host's resources, hormonal systems, behavior, and morphology have all been modified to serve the parasite's ends. The parasite's shape, position, and even reproductive timing are determined by the host's body. They are no longer simply two organisms in the host-parasite relationship. They have merged into a single functional unit. The rhizocephalan is a reminder of how incomplete our knowledge of marine life forms remains. New species of this group are described regularly, particularly from undersampled regions and hosts. Many deep sea and tropical crustaceans have never been examined for rhizocephalan infections.